A very warm welcome to one and all. Today our group is going to give a presentation on the topic of leaf, under which we are going to cover morphology of leaf under the subject of pharmacognosy and phytochemistry 1. We all are well known what a leaf is and how it plays a great role in giving oxygen to us. So today we are going to talk about leaf more deeply in the morphology terms. A leaf, also known as phylopodium, is a lateral, generally flattened structure borne on the stem. It develops at the node and bears a bud in its axil. The axillary bud later on develops into the branch. Leaf originates from the shoot apical meristem and are arranged in an acropetal order. Vegetative organ for the photosynthesis. Photosynthesis, as you all know, is a process by which plants use sunlight, water and carbon dioxide to create oxygen and energy in the form of sugar. Now you can observe the main parts of the leaf. It consists of a leaf base, petiole, lamina, bud axillary and stipule. Parts of leaf. There are three main parts of the leaf base, petiole and lamina, which you observed in the previous slide. The leaf is attached to the stem by the leaf base and may or may not bear two lateral small leaf-like structures called stipules. Petioles help in holding the lamina to light. Long, thin, flexible petioles allow leaf lamina to flutter in wind that helps in cooling of leaf and bringing fresh air to the leaf structures. The lamina is the green expanded part of the leaf with veins and veinlets. There is a middle prominent vein called midrib. Veins provide rigidity to the leaf blade and act as channels of transport of water, minerals and food materials. Now we will talk about venations. Venations are the arrangement of veins and the veinlets in the lamina of leaf. They are of two types, reticulate venation and parallel venation. When the veinlets form a network, the venation is termed as reticulate venation, which are observed in the dicot plants. When the vein runs parallel to each other within the lamina, the venation are known as parallel venation, which is usually seen in monocot plants. You can see the difference between the reticulate and the parallel venations. Now if we talk about the types of leaf, there are two types of leaves, simple leaves and compound leaves. You can differentiate it with the help of diagrams. In the simple leaf, there are single and undivided laminas. And if we talk about compound leaves, leaf lamina divided into many leaflets attached to the mid vein and having its stalk. They are again of two types, pinnate compound and palmate compound. In pinnate compound, leaflets are present on a common axis, while in palmate compound, the leaflets are attached at the common point at the tip of the petiole. Now if we talk about phyllotaxy, phyllotaxy is the arrangement of leaves on the stem or branch. It is of alternate, opposite and whorled. Alternate, single leaf arises at each node in alternate manner. Example, China rose. Opposite, a pair of leaves arise at a node and opposite to each other. For example, Guava and Calotropis. Whorled, more than two leaves arise at the node and form a whorl. It is seen in Allostemia. You can see the alternate, spiral, opposite and whorled. Phyllotaxy. Now we will talk about the modification of plant. Firstly, the question arises, why do plants modify? In many plants, leaves get modified to perform some special functions other than the normal ones, such as photosynthesis and transpiration. Sometimes these modifications are in response to certain environmental conditions. Let's take a look on some examples of the modification of leaves. First, tendril. Tendril is a modification for climbing purpose, which is seen in peas. Next, spine. Spine are the modification for the defense purpose, which is usually seen in the cactus plant. Storage of food, fleshy leaves of onion and garlic, leaves of certain insectivorous plant like pitcher plant, Venus flytrap are also modified leaves. Reduced leaves in Australia acacia, the leaves are small and short-lived. In this case, the petiole expands and become green to perform photosynthesis. You can see the modification, tendril, spine and storage. Now the chemical test for leaf. Leaf starch test. Leaf is put into the boiling water and then boiled leaf is put into the boiling ethanol, which decolorizes the leaves. Then decolorized leaf is put into the cold water and then leaf is taken into the Petri plate and 1 drop of iodine solution is added. You can observe if dark blue-black color observed, which means it has given positive result, which means starch is present in the leaf. Whereas if no color is observed, This means that starch was absent in the leaf and it gives negative leaf starch test. Now we'll talk about some of the medicinal uses of leaves. Medicinal plants having medicinal uses. First is Ocimum tenui florum, commonly known as holy basil or tulsi, is an aromatic perennial plant in the family Laminaceae. It is widely used as a herbal tea, commonly used in the Ayurveda. Tulsi plant is a native to India subcontinent and grows throughout Southeast Asia. If we talk about the kingdom, Plantae, phylum, and